Today I would like to show you three wonderful little machines that copy and translate our DNA. There are millions of these in every cell in our body, and also in every bacterium and in every plant. I will assume that you've all heard about DNA, that it carries a code, and that small differences between us in our DNA is what makes us unique. But when you see how intricate and beautiful these little machines are, it will fill you with a sense of awe and wonder, and you should say, where on earth did these come from? Clearly, either they were invented by the ancestors of the bacteria, or they were invented by the Creator and given to the bacteria, from whom we have all inherited these machines, and so have all the plants and animals. One of the most amazing things about DNA is how compact it is as a store of data. For instance, inside the head of a sperm, the space available is only about 1/10,000,000th of a cubic millimeter. But in that there is a whole meter of DNA with enough information to build a whole human being. Our genes are made of DNA. Thousands of genes are joined end to end along each chromosome, as I've shown in this picture. Now, most genes carry coded instructions, a recipe for how to make a protein. And when you put them all together, the 20,000 recipes for 20,000 proteins, you can build a baby. We have about 20,000 different proteins in each of us. They're all essential, and they're each one doing a special job. If any one of them is missing, we would probably not be alive, or we would have an inherited disease. At the top in the middle, you can see there are 6 proteins arranged in a ring. Now, there are many examples of this in us because these are the gates that allow or disallow molecules to come in or out of the cell. Here are 4 more. The second from the left is hemoglobin, which, as you know, carries oxygen around our bodies. Antibodies are made of 2 long and 2 short proteins, and between them they form a binding site for pathogens like viruses. This shows a ring of identical proteins that form a rotor of an electric motor that makes fuel for the cell. Different proteins have different shapes, but they all start off as a chain, like a chain of beads. If you look at this carefully, you can see that the beads have got different letters. In fact, there are 20 different sorts of beads that can be put together to make a protein. The beads are actually molecules of amino acids. The protein in the last picture was made of about 450 beads. This is the longest protein that's yet been discovered. This is titin in your muscles, and this is 34,350 amino acids long, and it's divided up into 6 springy bits. Here is a tiny bit of heart muscle or arm muscle, and you can see the titin on the right. It's that yellow springy thing. If it were not for this, our muscles would all be floppy and we'd have great difficulty moving and our heart wouldn't beat properly. And this is an essential enzyme found in bacteria, plants and animals, and us of course, and if any of the amino acids is in the wrong order, it just won't work. So all proteins are made up like a string of beads from those 20 amino acids shown on the left. Vegetarians among you will know that there are 9 of those that we have to have in our diet because we can't make them ourselves. And the other 11 we can make. The large ball in the middle of this picture is one nucleus in a human cell. You could get 100 of these side by side in 1 millimeter. Now, the nucleus is where all the DNA is, pretty much. But there's two things I'd like to make sure you've got before I can carry on. One is that the code to make the proteins is there in the nucleus, but the protein factories are outside the nucleus. They're the very small black dots that you can see, tens of thousands of them. Not the little circles, that's something else, but the protein factories are the tiny black dots, and somehow the code has to be got out of the nucleus to the factories. So what actually happens is that the code is copied onto a messenger which carries it out of the nucleus and finds itself a little factory. And the other thing is that we all start off as one cell, a fertilized egg, which divides into two, and each of those divides into two, and so on, until you end up with trillions of cells before the baby is born. Now, every single cell has got to have a complete message system. All the DNA has to be copied, therefore, so that every cell has got a complete set of instructions. Here's a tiny bit of the DNA in one cell. This is a small piece of one chromosome. There is about 6 feet of it altogether in every cell, and in fact, since you have about 100 million cells, the DNA in your whole body would reach from here to the sun and back about 60 times. And this is all packaged up inside the nucleus of each cell. Whether you take DNA from a bacterium, or a fish, or a human being, or a tree, it all looks the same. It's rather boring really, except for the code that it's carrying. You can see the code is actually in between the spiral bits, those coloured molecules in the middle. Now it's possible to uncoil one of these strands of DNA, and then it can be unzipped and expose the code so that the code can be copied, as you will see in the next video clip. This is the famous model of the structure of DNA which Watson and Crick discovered in 1953. We can come back to this later if you want. In this coloured model of DNA, you can see quite clearly that there are 2 strands coiled together, and they're held together by pairs of molecules which there are coloured yellow, green, blue and red. And there are 4 kinds of building blocks that make these connecting molecules: A, T, C and G. Now, if you imagine uncoiling that, it would look something like this. And here you can see even more clearly that the blue ones pair with the yellow ones and the red ones pair with the green ones. Now, if you imagine unzipping that and then supplying thousands of new coloured building blocks, what could happen? Well, you should be able to see that a new top strand would be formed from the bottom strand. It would template it off, and a new bottom strand would be formed on the top strand. So both sides would form a copy of their previous partner. So you start off with one double DNA molecule in the double helix, and you end up with 2 double helices. So the DNA has been copied. There's the coiled DNA. If you uncoil it, it can be unzipped, and the yellow things in the middle are the bits carrying the code. And here in action is this incredible machine that copies the DNA. You have countless numbers of these machines in your body, working day and night as your cells grow. Now you can see the DNA is coming in on the left at the bottom. It's unzipped, and then half of it is going across to the right, and a new template is formed to produce another double helix. But what's going on at the top of the picture is a bit more complicated. This is an accurate representation of what's going on because it's based on our knowledge of the structure of the components of this machine. This model will help me to explain what's going on. The DNA to be copied is coming in at the right, and the two copies that are produced are coming out at the left. Now, if you look in the middle, you can see a blue triangle. Now, that is unwinding the DNA and unzipping it. And if you look at the bottom half of the diagram, you can see the strand with the green building blocks is coming along from the right to the left, and at the bottom there's a yellow box which is adding yellow building blocks to the green ones as it zips its way along from the left to the right. The complicated thing is that the two strands have to be read in opposite directions. So the yellow box at the top, which is doing the same thing, namely in this case adding green blocks onto the yellow building blocks, it can't read from left to right, it has to read from right to left. So it starts just after it's unzipped and it moves along to the left. But then it has to go back and start again on the next bit. So the result is that you have great loops of the DNA being copied, sticking out from this apparatus, and then they have to be put back together again and joined up. I think it's worth having another look at that bit of film. The DNA is coming in from the left, and on this bit you see the blue, that's the protein which is unzipping and unwinding the DNA, and half of it is going across to the right to produce a new strand. The one at the top is the one that has to be produced in loops and separate sections which then have to be joined back together. This whole machine is the same in bacteria and in animals and in plants, which means that either bacteria are our ancestors or the grand designer decided to use the same machinery in all living things. So that's one incredible machine down and 2 to go. Now, do you remember I mentioned earlier that the instructions for making the proteins to make a human being are in the nucleus, in the DNA. But the protein factories are outside the nucleus. So somehow we've got to get the coded instructions copied so that they can be carried out of the nucleus and find a protein factory. Now, if you look at the top, that is called the template strand. That's the DNA code. And that is copied onto a messenger RNA molecule. And then that is translated outside at the protein factories. Now, there's two things to notice here. One's not really very important, and the other one is important. So the first thing is, you may have noticed already that if you look at the RNA messenger, there's a new letter there. That's U. Instead of T, it's U. That's because RNA, instead of having T, has got U in its structure. But that doesn't really matter because the code is still faithfully copied onto the messenger molecule. And the other thing to notice that is important is that on the messenger RNA you can see that there are 3-letter words. The first one is AUG, and the second word is CCG. Now, there are 4 letters in the RNA code, A, U, G and C, but the message is written in 3-letter words. Now the mathematicians among you will know that you can actually write 64 3-letter words if you've only got 4 letters to use. And in fact all 64 are used. 3 of them actually mean stop, which means that when the factory runs across the stop codon, it stops making the protein and it's peeled off. But the other 61 are all used to make a particular amino acid. So at the bottom you can see the first 10 amino acids in a protein chain, which is faithfully representing the code at the top, which is the beginning of the message for how to make that protein. Here then is the second of my incredible machines. It's an enzyme complex that is copying the code from the DNA and making a messenger to carry the code out of the nucleus. So there are 2 entry channels and 2 exit channels. The DNA is coming in from the right hand side. Inside the polymerase it's split into 2. One half, the orange half here, is the template strand. That code is copied onto the messenger RNA. The green strand that's coming out on the left at the bottom, and then the DNA is wound up again and comes out at the top on the left. Now the other entry channel is for all the building blocks which are needed to make the messenger molecule. There are 4 sorts of building blocks, one carrying an A, one carrying a U, one a C and one a G, and they're all assembled according to the instructions. This is the actual shape of the machine. It moves along very fast along the DNA, and out comes the messenger, which in this case is yellow. And here it is in real life. There is a gene starting just to the left of the middle and going all the way along to the right hand side. And on it you can see lots of those little black dots in a line. Now those are all the enzyme complexes which are copying the code onto the messengers. They start on the left at the top of the Christmas tree, and they work their way along towards the end on the right. And as they do, they are churning out the messengers. So on this one gene you can see maybe 100 messenger RNA molecules being made, and when they're finished they break off and they can be carried out of the nucleus into the cytoplasm. When a particular protein is required in a cell, the gene which carries the code to make it is switched on. Then this machine comes along and then with a supply of the 4 different building blocks can make a copy of the message from the template strand and make a messenger molecule to carry the code out of the nucleus. Now we come at last to my third and most unthinkably beautiful machine. I say unthinkable because it must be 60 years ago that Francis Crick suggested that there must be a machine which can translate the code into protein, but it was so difficult to imagine how that might possibly be done that it took many years before this was worked out. And I say beautiful because, well, it's the idea that this is possible at all is so beautiful. Well, to do the translation you need 2 things really. You need the green factory in the middle. There are hundreds of thousands of these in each cell, all the same, and the 2 halves of this factory fit together neatly around the messenger molecule, which you can see a bit of going across from the left to the right. The other thing you need is those yellow molecules shown in the middle, the things that look like the old-fashioned sardine tin openers, if you remember those. Now, those transfer molecules, there's 31 different transfer molecules in humans, and at the top they all carry an amino acid, which is one of the building blocks for the protein, and at the bottom, notice they've all got a 3-letter anticodon, which only fits into one position on the messenger. Can you see, for instance, on the right, the anticodon on the transfer on the right-hand molecule is UGC, and that only fits onto the codon on the messenger ACG. So there's no ambiguity in this. And when the molecule on the left moves across to the middle, its amino acid is joined onto the one on the right. Then the one on the right is released and the new one comes in on the left-hand side. Here is one particular transfer molecule. They all look quite similar, but they're all unique. At the bottom of each one there is what's called an anticodon, in this case CGG, and that fits only one of the codons, in this case GCC. And at the top they all carry a particular amino acid. Here comes the messenger out of the nucleus into the cell, ready to be translated. It has first to find the protein factory, that's the ribosome, which attaches to the beginning of the message. And then it has to be translated. Now, these transfer molecules are the ones that actually do the translation. I've mentioned before, at the bottom you can see the 3 bases that make the anticodon. That determines where it fits on the message, and at the top it carries one amino acid. They come in one at a time, and then the amino acids join together to make the protein, which comes out at the top. What an amazing mechanism it is. And here is the real thing. Along in a U-shape is the messenger RNA, and all those black bodies are the ribosomes, which are walking along it. And as they do so, they start at the top right-hand corner, and as they move along, they begin to make their protein molecules, which in some cases you can see sticking out from the ribosome. So if you follow it round until you get to the top left-hand corner, there you can see the protein threads coming out of the ribosomes are quite long. So there's a start point and an end point, and at the end the proteins are released from the ribosome. So the human body is built from more than 20,000 different proteins, and Nearly 20,000 different genes carry the instructions for making those proteins. And every cell has to have a copy, so the DNA has to be replicated. But the copy of the instructions has to pass out of the nucleus into the protein factories, and that's why you need the messenger RNA molecules. And lastly, the instructions can be translated into protein molecules.